Hi class! So you might notice this is not at school, this is being recorded at home. My computer kind of died halfway through lecture today, so we're here. We can go through it a little bit more quickly than normal, partially because there's not going to be student questions or things like that. So here's where we are from today. And you do have, this class is going to get done a little bit early, so do go and take your quiz. Do please take your quiz. Also remember to do your homework before Sunday midnight, and hopefully October's treating you okay and things go well. So we're here to finish up the last little part of cellular respiration. So, what we've been talking about this whole week, Monday and Wednesday, was all about how, okay, we start with glucose, and in the presence of oxygen, we're going to break it all the way down, and we're going to get energy off. We're going to harvest energy that's stored in this organic molecule. And that works great, usually. The problem is the electron transport chain needs and really likes having oxygen, because otherwise, as the electron transport chain accepts electrons, things go okay. Once the electron moves through the electron transport chain, it gets to the end, it needs to leave. If it doesn't leave, the whole thing breaks because you just clog up the thing with electrons then. You have to have electrons leave for new electrons to be able to fit. And what picks up those low energy electrons at the very end is oxygen in regular cellular respiration. You have, in aerobic cellular respiration, oxygen picks this up and it can just keep the cycle going. If you don't have oxygen, you need some other things to happen. So most regular cellular respiration, you need oxygen to produce this ATP. If you don't have oxygen, you're going to need to do some other way of getting this done. So one version is to do this. It's called anaerobic respiration. So it's respiration that's going to use an electron transport chain, but it's going to have something other than oxygen pick it up. This isn't super common. I mean, it happens, but these little purple sulfur bacteria can get it done. If you want to briefly watch a video about this, they're kind of cute. These little guys here, they work. The system works very similarly to how it does in just a regular electron transport chain, which is probably less surprising if you look at, say, the periodic table right here. You can see oxygen's up here. Sulfur is very chemically similar to oxygen in many ways, and so it's electronegative as well. They can pick up those low energy electrons at the end of the electron transport chain, and it can just keep going. So again, you're not going to have this thing happen to you. You're not using anaerobic respiration with sulfur, but some little bacteria buddies can get that done. Okay, so that's a side note. What's more important for us right now is looking at something called fermentation. So you've seen this picture before. This was the first part. This was glycolysis. So when you're going from glucose all the way carrying it through glycolysis, and then that citric acid cycle, right? So pyruvate oxidation, citric acid cycle, and then taking it to oxidative phosphorylation, that whole way. The first part you start with is fermentation. And you might remember, you get a tiny bit of ATP out. So you pay 2 ATP in, you get 4 ATP out. Glycolysis, this first step, actually gave you a little bit of energy. You were able to harvest a little bit of the energy from the glucose into ATP. So cells can run this to get energy. It's doable. You'll notice there's no molecular oxygen required here at all. You don't need to be in an oxygen-rich environment to get this done. And this can just run. it isn't as efficient as the whole just aerobic cellular respiration, right? Taking it the whole way. If you take this the whole way, glucose can give off like 32 molecules of ATP. From glycolysis, you can get a net gain of 2. So it's much less efficient. But you don't need oxygen. So there is that. What happens here, right? There's this glucose, pyruvate plus water, giving off a little bit of ATP. You'll see also, to have this happen, you need to have NAD+. So you need to have NAD+, which are these, they're empty electron carriers, right? So they're empty, and they pick up high energy electrons from the glucose, right? And then they're full, right? Because they've got high energy electrons in here, and they're good to go. In order to keep running this, in order to keep having this first part happen and being able to just get energy with this, you need something to empty out this electron carrier. If you were doing aerobic cellular respiration, these electrons would be dumped off at the electron transport chain. If you're not using the electron transport chain because you can't, because it's clogged with electrons, because there's no oxygen to clear it, you need something else to clear this of electrons so you can keep running this. So, the way to get this done is called fermentation. There are two main types or flavors of fermentation, I guess. The end point for all of them is basically it's glycolysis plus some reactions that are going to empty out this NADH, empty it back out to NAD+. So it comes in two types. It comes in alcohol fermentation and lactic acid fermentation. So alcohol fermentation, it's going to make alcohol. This is the alcohol that you use. This kind of fermentation is what puffs bread up. Yeast does this. It happens wine making. It happens all over the place. All right. When alcohol fermentation occurs, you've gone from glucose. You had the first glycolysis step. You've broken it down to pyruvate. Then alcohol fermentation takes pyruvate. It does several things. What it does first is it breaks off carbon and 2 oxygens, and that leaves as carbon dioxide, which is why bread puffs up, right? So the yeast is doing its thing, right? Alcohol fermentation is happening. One of the byproducts of that is carbon dioxide production. So it puffs bread up, right? The second step produces ethanol, which is an alcohol. So like, if you smell beer, you smell that really yeasty smell. You're smelling, or if you smell bread and it smells kind of like beer, you're smelling that ethanol, right? The other version is lactic acid fermentation. This kind doesn't have alcohol as byproduct and doesn't have carbon dioxide as byproduct. What it does is you've done the first glycolysis portion, you made it with pyruvate, and then you give the electrons back to this pyruvate thing and you end up forming lactate. Lactic acid is the stuff that it shows up a lot in things you eat. You taste in yogurt. You can taste that like tangy taste. You're tasting lactic acid made via this way. And so you're a lot more similar to yogurt than you are to bread in that human muscle cells will use that lactic acid fermentation to generate ATP. If oxygen is scarce, they do this all the time. In some ways, this is really, really good, right? One problem is if you used alcohol fermentation in your muscles, if you worked out, you'd accidentally get drunk because you'd be producing alcohol inside of you. So that doesn't happen, that's good. But while alcohol doesn't build up in this, right, alcohol can build up in alcohol fermentation. Alcohol doesn't build up in lactic acid fermentation. what does build up is lactic acid. And lactic acid buildup is thought to be responsible for certain types of sore muscles. Also, using lactic acid fermentation is not nearly as efficient as using regular aerobic cellular respiration. But you can do this even if you're short on oxygen, right? So that's helpful. So here's the overall pictures, lots of squiggly lines, but what we're looking at here is this one on the left is the alcohol fermentation. I realize I'm not even, I am recording, okay. Not in class, I feel like I have to check at least once that I'm recording. Here, I feel like now it's just a habit. Okay, so we went from glucose to 2 pyruvates, which just the regular glycolysis portion, right? Generated 2 net ATPs, beautiful. We have 2 of these NAD pluses, they pick up electrons, and now they're full, right? This was the glycolysis portion. In order to regenerate NAD plus, we need to get the electrons off of this. So here, pardon. In alcohol fermentation, this pyruvate is broken down. Get rid of this carbon dioxide. Have this little molecule here. This molecule picks up those electrons, ends up being ethanol, so alcohol, and then you're back to start the cycle again. So you can keep running glucose through indefinitely. You can keep having this happen. You don't need oxygen. You do make ethanol as a byproduct, and you make carbon dioxide as a byproduct. Great. That's alcohol fermentation. Lactic acid fermentation is very similar. You're running glucose through this first part of glycolysis, right? You're producing ATP. You're filling up these electron carriers, right? But then you're using this pyruvate. You're using that to pick up the electrons. So as you pick up the electrons and empty this back out, you're forming lactate. And this can keep running. You're not making carbon dioxide, you are making lactate, and this builds up. Great. So far so good? I hope. This video is in your slides. Go ahead and watch it. Actually, I guess we could watch it right now. We will watch it right now. It's not the world's most interesting video, but it's worth seeing.
All cells are able to synthesize ATP via the process of glycolysis. In many cells, if oxygen is not present, pyruvate is metabolized in a process called fermentation. By oxidizing the NADH produced in glycolysis, fermentation regenerates NAD+, which can take part in glycolysis once again to produce more ATP. The net energy gain in fermentation is 2 ATP molecules per molecule of glucose. Fermentation complements glycolysis and makes it possible for ATP to be continually produced in the absence of oxygen. There are 2 types of fermentation. Alcohol fermentation, which occurs in yeast, results in the production of ethanol and carbon dioxide. Lactic acid fermentation, which occurs in muscle, results in the production of lactate.
Great. So hopefully we all kind of saw that. You start with this 6-carbon sugar, right here. You break it down into 2 pyruvates. You get 2 ATPs off of it. You get some of the electrons out here. Fermentation, either, depending on the kind of organism you happen to be, either runs that through lactic acid fermentation and keeps the 3-carbon sugar, but just puts some more electrons on it different ways, or breaks down even farther into ethanol and carbon dioxide. You'll note that this is all happening outside mitochondria. So even if you're something that doesn't, sorry, my computer is making beep sounds. Even if you're something that doesn't have organelles inside of you, some sort of cell, this process doesn't require organelles, doesn't require mitochondria. Great? Cool. So, in general, this whole overall process, fermentation and anaerobic and aerobic respiration, they're all going to use this first glycolysis portion. They're going to oxidize glucose, they're going to harvest chemical energy from food. They're all going to use NAD+, to grab the electrons during glycolysis. But where it gets different is what you do next. So if you just have regular NADH, it's got electrons there. In fermentation, you're using an organic molecule to clear those electrons. In cellular respiration, you're transferring those electrons to the electron transport chain. Let me plug my computer before it completely dies, because if it dies again, I'll be super sad. This morning it didn't die because of power, it died just because my computer has special challenges. By special challenges, I mean I've had it for like way too many years. Okay. And so yeah, we saw this before. Cellular respiration produces way more ATP per glucose molecule than fermentation does. But then you still have to, use oxygen. But okay. Thus far we've been acting as if glucose is the only source you can use for some sort of organic fuel. Like, that's it. And that's not the case. You know that while you can eat glucose straight, or you can have a diet that consists primarily of, those little gummy bear things, that's not really what you're doing. You're doing other things too. And it probably doesn't surprise you that you get energy from other sources too. So, good news, the rules are basically the same, it's just where things come in are a little bit different. So, glycolysis and the citric acid cycle interconnect with a whole bunch of other pathways. So, catabolism, the ability for the cell to break down complex molecules into smaller bits, you can do that with lots of different things. Lots of words here for you to study. This is the picture we're looking at. Main types of things you're eating are going to be proteins, carbohydrates, and fats, right? So carbohydrates could be something like glucose, right, which we've seen so far, or it could be something more complicated like starch, right? You can have, or you can have something in between. You can have something like sucrose, which is a more complicated sugar than glucose, but it's still, sugar stuck together. You can also have proteins, which are a bunch of amino acids strung together, and then fats, which are glycerol plus fatty acids, right? Okay, so these are the kinds of things we're eating, whether that's going to be potatoes, or peanut butter, or margarine, or whatever it is you're eating. Sugars are going to go into this energy harvesting mode. You expect it to come into glycolysis. Simple sugars you're going to rearrange and get in just as glucose. You can also have them feed in slightly later, partway through the steps of glycolysis, depending on what type of sugar it is and what's been adjusted. Right? Amino acid, so pyruvate's this 3-carbon chunk, right? Amino acids have carbons. You can get this pyruvate chunk from amino acids. You do have to get rid of this nitrogen bit, and getting rid of nitrogen is actually a really big deal for most organisms. Figuring out how to do that is important. But you can have these amino acids, you can break them up and feed them in right here. From this, you don't get the substrate level ATP that was generated by this. That's okay. Who cares? You're going to get plenty of energy back. Not a problem. Fats you can also feed into this, so the glycerol portion can come in partway through this glycolysis stage. You can also keep going. So proteins, you could feed some in here. You can also feed some into this. You can have it come in as acetyl-CoA. Same deal with fatty acids, right? So this tiny carbon that are connected to each other with the acetyl portion of acetyl-CoA, that you can have gotten from fatty acids or amino acids, right? So you're funneling in these organic molecules through a variety of ways into this energy harvesting system. Now proteins, you can also feed directly into the citric acid cycle. You can feed it right on in. So all of these molecules are coming in via a bunch of different pathways, but they're all converging on the same pathway. At the end of the day, you're expecting everything to show up at the citric acid cycle, and then to go through, whoa, I don't know what happened. There we go. Everything to go into the citric acid cycle, right? And then you finish breaking up these carbon-carbon bonds, and you release carbon to carbon dioxide, and you harvest these high-energy electrons, and then you use those electrons to go, they go into the electron transport chain. That's going to drive hydrogen ions across the membrane of the mitochondrium. Then those hydrogen ions are going to be able to fall back through ATP synthase, and then you'll be able to synthesize ATP. Right? Whew, lots happening, right? Great. So that's just to be honest, there are other things happening too. But classically, you look at it through glucose, because those lines are just beautifully straight. Now, this is all well and good. This is great. Your cells, other people's cells, cells of all sorts of things, are very interested in being able to harvest energy from other sources and use it. And that's important, right? Being able to harvest energy, that's critically important. But you don't want to go overboard. So your cell wants to use its proteins, carbohydrates, and fats for fuel. But it doesn't want to make more ATP than it really needs. There's no point to keep breaking down carbohydrates if you don't need them. There's no point to destroy a bunch of proteins if you could be using them for something else. There's no point to be burning fat here if what you could be doing is to be storing it, right? From the cell's point of view, it's just very wasteful. So they've got some controls on this, right? You can kind of imagine, well, we'll look through here. This thing, lots of errors going on, I recognize that. But this is showing the control of the controlled energy production. So remember enzymes, right? How enzymes can catalyze reactions they can make so that they can happen in a biologically meaningful time frame. This whole process of cellular respiration uses a whole series of enzymes, right? So if any enzyme goes, you're not expecting the process to happen. One of the first enzymes used in this process is this thing called phosphofructokinase, right? It's this enzyme that's really important in one of the first stages of glycolysis. We briefly saw it as just, oh my goodness sakes, my computer's definitely having problems. One of the first steps of glycolysis as it's trying to get glucose prepared to carry on and be changed into pyruvate. Well, if the cell has a bunch of ATP floating around, right? There's plenty of ATP. This ATP acts and it inhibits this enzyme. It's an inhibitor of the enzyme. You can imagine, like, if you're making pancakes for somebody, right? You're there making pancakes, it's great. If you see a huge stack of pancakes developing, right, because you're just going for it, you're doing a great job. If you've got a really big stack of these, having all those pancakes should inhibit you from making more. You're like, okay, I don't need to make any more, we have a bunch. I'll hold off. This ATP is physically inhibiting this enzyme here. So lots of ATP, you don't run this process as much. If the cell uses a bunch of ATP, and it's now gone, and you don't have ATP, this enzyme's not being inhibited by that. So this enzyme's not being inhibited, so this whole process is just chugging along beautifully. Does that make sense? I hope that makes sense. Similarly, AMP stimulates this enzyme. So ATP, this TP portion stands for triphosphate. There are 3 phosphate groups attached. You've heard of ADP. We see it back all over the place. We see it over here, right? ADP plus phosphate ATP. ATP stands for diphosphate. There are two phosphates. AMP stands for monophosphate. And this is ATP that's lost two of its phosphates, meaning the cell's kind of hurting for ATP. Like it's burned through a bunch of it. So if you have a bunch of AMP where you don't have ATP, you've used a lot of ATP, the cell doesn't have what it needs. This AMP encourages this enzyme and gets it to go better. Similarly, just to make sure that this doesn't turn out more ATP than is necessary, the citric acid cycle, the first thing in it that it makes is something called citrate. And there's a whole bunch of citrate in the cell. It acts to inhibit this enzyme as well. So it's just ways that the system controls itself, right? So the system is going to be producing ATP, and as it produces ATP, it's going to turn the system off, right? So then in the absence of ATP, ATP is produced, and in the presence of lots of ATP, it's not produced. So hopefully that makes sense. These things are questions worth knowing, things to think about. Try to answer them yourselves. If you can't, look it up in the book, but do try to at least think about it. Like, okay, what is oxidative phosphorylation? What is fermentation? How can I understand the difference between these things? That's what we've got. I hope you have a wonderful, wonderful weekend. Do remember about the quiz and your homework. And yeah, I'll talk to you all later. Bye.